Welcome back. Today we are going to be talking about organic chemistry, which we call the chemistry of life. We've already reviewed some inorganic chemistry. And organic chemistry just deals with compounds that contain carbon and hydrogen, which are the elements that really build up the different parts of living organisms. So when we talk about organic chemistry, we're mostly talking about what we call biomolecules or biological molecules. And these are the molecules that are present in organisms that are essential to at least one cell process, usually many. And they are macromolecules, so kind of larger. They are made up of carbon, and they are built out of what we call monomers. Those are the subunits. And so we hook all those monomers together into what we call a polymer. And if you remember your Latin roots, we can look over here, and mono means one and poly means many. And in this case, mer is meaning the subunit. So if we just have a monomer, that is one subunit, when we hook them together, we have what's called a polymer, or many subunits linked together. And that's how we make biomolecules. So because carbon is the most important part of our biomolecules, I just wanted to review a little bit about what we've learned about carbon already. So carbon can bond with as many as 4 other elements, and remember that's because that outer valence shell has 4 electrons, and we want to be able to obtain the octet rule, so it would like to have 8. And it typically bonds with hydrogen, nitrogen, oxygen, phosphorus, and sulfur. And those, remember we talked about those before, those are our major elements within the body. And so one thing to know about carbon is that usually a carbon-to-carbon bond, I guess I didn't put that in there, it also bonds with other carbon atoms. And that carbon-to-carbon bond is very stable. So it can form long chains. And when we do, we call those long chains hydrocarbons. So a hydrocarbon is just an organic compound consisting entirely of hydrogen and carbon. So we can have long singular chains like the one on the left. And remember those single drawings there, those single lines indicate a pair of shared electrons. So we can have a long chain like that, or hydrocarbons can also form ring structures like these on the bottom right, our cyclopentane, cyclohexane, and our benzene ring. And notice that carbon not only can form these single bonds like we have here, or even in our cyclopentane and our cyclohexane, but it can also form double bonds like here in our benzene ring. And in addition, it can also form triple bonds. We've mentioned that before. So, but the double bonds and the triple bonds are not as flexible as our single bonds. So that determines the rigidity of our hydrocarbons. So when we're looking at carbon, it can typically form 3 major structures. And so we've kind of talked about those with the hydrocarbon. So we have either a straight chain, a branched chain, or a ring structure. And all of that is just going to depend on the carbon-to-carbon interactions. So the carbon chains that are created by linking carbon to carbon are what we call the carbon skeleton, or the backbone of the organic biomolecules. And they start to create the shape. But then we add what we call functional groups to make a huge variety of organic molecules. And most of the chemical reactivity of a biomolecule is dependent on these particular functional groups. So in our chart here, let me just get my little pen. R is just going to stand for our carbon chain, or that carbon backbone. And we can add any of these functional groups. So if we add an OH on the end of our carbon chain, we have what's called an alcohol. And y'all are familiar with that. Propyl alcohol, propanol, that's the same thing. We can add, let's say, I'm trying to think of what, an aldehyde group. So this right here is our aldehyde group, and that means this particular one is ethanol, so aldehyde. We can add a carboxyl group. That's very common, and that usually gives us an acid. So in this particular one, I think we have, what do we have? Ethanoic acid, or carbonic acid, or carboxylic acid. Any of those, we can make those. And then we can also add basically the carboxyl group without the H. That creates an ester. So all of those determine how our biomolecule interacts with other molecules, so the chemical reactions that take place. Now, you're not going to have to know all the different functional groups, but I want you to be able to understand when I give you like this particular formula here, that you would know that the R group is our carbon chain, and so there's 2 carbon chains here, and then that the other part would be a functional group. Okay? I don't need you to know the names of each functional group, but I need you to recognize the formula that shows where the functional group is. Okay, so how do we make these biomolecules or synthesize them? The cell uses a dehydration reaction. It's also called a condensation reaction, where it takes the subunits and forms a larger structure by linking the monomers and getting rid of a water molecule. And what we mean by monomers is basically smaller subunits of something. And that's going to be different for each type of biomolecule, and we'll look a little bit into that as we talk about the different ones. So this is a great little flow chart here from your textbook, just to show how we make these. So the synthesis, as you can tell, is we are getting rid of water and bringing together those monomers with this bond right here. Conversely, if we want to break apart our biomolecule, we're going to do what's called a hydrolysis reaction. So hydro meaning water. So we're going to add water to break apart that same bond. So we have an OH group on one end and an H group on the other, linked to the 2 separate monomers, and that breaks that bond. So that's how we synthesize and degrade our biomolecules. So just something else that you'll need to know is what we call isomers. And isomers are organic molecules that have the identical chemical formula, but different shapes. And remember, we talked about how shapes or structure informs us about the function of a protein or of a particular biomolecule. Sorry, not just proteins, everything. So in this particular case, our chemical formula is C4H10. So we know we have 4 carbons and 10 hydrogens. Well, in the first one, we have this straight chain, just very easy, and that's the shape of butane. And then with methylpropane, we still have 4 carbons and 10 hydrogens, but they're arranged differently. So this up here comes out, and look at the shape of the molecule over here. So that those serve different purposes within living organisms, but they have the same chemical formula. So our biomolecules fall into 4 major categories, and that's carbohydrates, lipids, proteins, and nucleic acids. And we categorize them based on their chemical structure and on their function. So you should have a chart that is labeled biomolecules. You're going to want to pull that out about now so that you can compare and contrast these 4 types of biomolecules. Okay, so our first type of biomolecule are our carbohydrates. And you're used to hearing that because people talk about eating carbs or trying not to eat carbs, depending on what their intention is. And so we find these oftentimes in breads, in fruit, that sort of thing. And carbohydrates are made up of carbon, hydrogen, and oxygen. And they're used typically for a quick source of energy for organisms, and then they also function in some structural roles as well. And these are made up of monomers called monosaccharides, which are sugars. Remember, our monomers are our subunits, and when we link those simple sugars together, they are called polysaccharides. Oftentimes you will see that the names of these carbohydrates end in OSE. In other words, sucrose, cellulose, fructose, those types of things. So those are our carbohydrates. So when we're talking about carbohydrates, and we said our monomer is a monosaccharide, so our example over here on the left are some monosaccharides that you might be familiar with. Our glucose, whose chemical formula is C6H12O6. Maybe I should write that down for you. C6. I am so bad at this part. H12O6. Maybe you can read that. That is what we use during cellular respiration, and it's broken down, and the energy that's released from that is stored as ATP. So this is our very important monosaccharide, glucose. Below it, I have two other very important monosaccharides, and they are ribose sugars. Notice that glucose has 6 carbons, 6, and then ribose is here in a pentose shape. It has 5 carbons. Ribose is the backbone of RNA, and deoxyribose, meaning without the oxygen, is the backbone for DNA. So those are some really important monosaccharides. Now, what we can do then is link a bunch of those monosaccharides together to make a disaccharide or a polysaccharide. And just like we talked about before, this is in a dehydration reaction, so we're removing water. So this is glucose plus fructose, and we've made this bond right here to join them together and make the disaccharide called sucrose. So we took 2 monosaccharides, glucose and fructose, bound them together and made a disaccharide called sucrose. When we link those all together and have a polysaccharide, we get things like starch or glycogen. One of our most important polysaccharides is what we call cellulose. And you can see down here at the bottom where you have a lot of glucose molecules all being linked together into these microfibrils, and then all of those come together to make cellulose. Why is that important? Cellulose is the main component of plant cell walls, and we're going to get into the differences between plant and animal cells in a little while. Makes me very excited. If you don't know my background, I am a plant person, but those cell walls are very important to plants because they don't have bones to keep their rigidity like you and I do, but the cell wall structure is what allows them to be strong and firm. So most of life on Earth is pretty glad that we have this specific polysaccharide, and it's actually the most abundant carbohydrate that we have on Earth. So another example of a polysaccharide that I don't have a picture of right here, but is the exoskeleton of insects or of crabs, for instance. There's something called chitin, and that is a polysaccharide that also serves, again, as a structural molecule that's really important, but that is in animal cells. So lipids are our second category of biomolecules, and lipids are composed of carbon, hydrogen, and oxygen, just like our carbohydrates were, but they are linked into these fatty acid chains, and then there is a glycerol molecule attached to them as well. Glycerol being an alcohol. Now, because of this, they are nonpolar and insoluble in water. And they are typically used for long-term energy storage, so that's your fats, right? They are used to compose the cell membranes. Animals use them for insulation layers, and then they're also used to create hormones. And then they're also used by plants as a protective outer coating. So we're going to talk about those because there are 4 different types of lipids. There's triglycerides, phospholipids, steroids, and waxes. And so those are sort of subcategories of lipids. So let's start with triglycerides. Just as the name would imply, tri being 3, triglycerides are made up of 3 fatty acids and a glycerol molecule. And when we go through the dehydration synthesis reaction, we combine those together into a triglyceride. And these triglycerides, or fats, are then stored in animals in what we call adipose tissue, or your fat tissue. And that is then used as in an insulated layer, and then can be accessed for being broken down to be used as energy when the carbohydrates have been used up. So there are a couple of different types of triglycerides, and you hear about this a lot if you are interested at all in nutrition. So we have unsaturated and saturated fat, or triglycerides. So our saturated fatty acids means that we have no double bonds. So if you look all through here, all of our carbons are linked with single bonds. And this typically comes from animal products, so animal fat. And that's where we get our milk and our butter, that sort of thing. Our unsaturated fatty acids are not saturated with hydrogen. That's what that unsaturated means. So you wind up with things like double bonds and triple bonds throughout the fatty acids. The reason that's important is because it causes kinks in these chains. And those kinks allow for those types of fats to be more fluid, whereas our saturated fats, see how they can stack up nicely? That allows them to be solid at room temperature. But our unsaturated fats have these kinks, and that's why you typically will see those in oils. And most of our unsaturated fats come from our plant bases. Our second type of lipid is the phospholipid, and just like the name implies, it is a phosphate group and a lipid. It's similar to triglycerides, but that third fatty acid that we had before is replaced with a polar phosphate group. And so that means that our third structure here actually has some polarity to it. And when it has polarity, that means that it can interact with water. Whereas the ones we looked at prior to, the triglycerides, they don't mix well with water. They can't interact with it because they are nonpolar. So now that we have a phosphate group, that phosphate group allows for interaction with water. But we still have a portion, which are the tails here, our fatty acid tails, that are nonpolar, which means they are hydrophobic. They don't like to be next to water because they don't have any charges to interact with water. This is why we talked about all that inorganic chemistry and knowing how chemicals interact with each other. It becomes important when you start looking at the cellular level. So phospholipids then, when they come together, tend to make these bilipid layers. And that is what composes the plasma membrane of a cell, is these 2 layers of phospholipids. We call that a phospholipid bilayer. And what that means is wherever the heads are interacts with water. So for a cell, that means that a cell that's usually bathed in some type of aqueous solution means that this outer phosphate group interacts with the outside of the cell as well as with the inside of the cell because the cytoplasm is made up mostly of water. However, the hydrophilic tails create this inside of the plasma membrane, and that does not interact with the water. So it is afraid of water. We call that hydrophobic. So we have the hydrophobic tails pointing towards each other, towards the inside, and then the phosphate heads pointed towards the outside and the inside of the cell in that bilayer because they can interact with water. Our third type of lipid is our steroids. And they are lipids that have 4 fused carbon rings to them. And I just put up 2 examples of ones that you would be familiar with. Cholesterol is a steroid. It's super important because it embeds in the plasma membrane that we talked about it a minute ago and allows it to be a little more fluid. Testosterone, obviously another important steroid in the human body. But notice that each one of these has 1, 2, 3, 4 fused carbon rings. Here's cholesterol's 1, 2, 3, and 4. And those 4 fused carbon rings are what defines a steroid. Our fourth lipid that occurs in nature is our waxes. And these are long fatty acid chains connected to carbon chains that contain an alcohol functional group. Remember, we talked about those functional groups being really important in how they interact biochemically. So usually if you're looking for an alcohol group, you're looking for that OH. And so we have these long chains connected to a carbon with an alcohol functional group. These are typically solid at room temperature. They have a very high melting point. And then they are hydrophobic. And remember, that word means that they don't like water. Typically, that means they don't dissolve in water. We see this in a lot of places. The probably easiest example that you can see is on a holly tree or a holly plant. The leaves have a very thick, waxy coating. And the reason for that is that they protect those leaves from sun as well as from evaporation. So if they have a waxy coating, that water is not going to be able to come off of the leaf and be evaporated very easily. Our third biomolecule is the protein. So proteins are actually very, very important in cells, and if you take just the dry mass of a cell, about 50% of it is made up of proteins, so that might give you an idea of how important they are. So within a cell, they're involved in transport and defense. They also play a support structure, and they are involved in all kinds of metabolism. So in fact, we call the particular proteins involved in metabolism, they're called enzymes. So if I say enzyme at some point in time, you'll have to know that that's a protein. Proteins are made up of amino acid monomers, and there are about 20 different amino acids that can come together in all kinds of different combinations to make up proteins. And sometimes it's easiest to think of that like the alphabet. You know, we only have 26 letters, but we put it in all kinds of different conformations in order to come up with various words that mean a whole bunch of different things. And it's very similar with proteins, and there are 20 different amino acids. So with those 20 amino acids being our monomers, we link them together through our dehydration reaction that we've talked about before, so taking out a water molecule to form a bond. And that's a covalent bond, and when we're talking about joining amino acids, we call that a peptide bond. So the peptide bond is a type of covalent bond. So if you'll remember, when we talked about covalent bonds, we said some are polar and some are nonpolar. Well, a peptide bond is polar, and the reason for that is because the oxygen is more electronegative than the nitrogen when they come together. And because of the polar nature of a peptide bond, hydrogen bonding is possible between different parts of the peptide. So the carboxyl group of one particular protein can bind with the amine group of another through hydrogen bonding. And this influences the shape and the structure of a protein. So the shape of a protein, or its structure, is really important. It determines how it functions within a cell. And there are 4 different levels, you could say, of the structure of proteins. And we call those primary, secondary, tertiary, and quaternary. So let's take a look at each one of those, and you'll start to understand why they're important. So the first level of structure for a protein is what we call the primary structure. And that's just the linear sequence of amino acids. And that linear sequence is determined by the information in the genetic code of each cell. So in this particular example, each one of these little pearls represents an amino acid. So that primary structure, just single chain of amino acids, that is our primary structure. The secondary structure of a protein is when the polypeptide starts to fold in particular ways, and it's unique to each protein. And there are 2 types of secondary structures of proteins. They can either be an alpha helix, which is down here at the bottom, or they can be beta pleated sheets. And some proteins just have secondary structure, up to secondary structure. Everybody has primary structure. Some stop at secondary structure. So for example, silk, as in this spider web, only has a secondary structure. It is composed only of beta pleated sheets. So it doesn't have anything further than that. Next is what we call the tertiary protein structure. And this is when we start to get the 3D shape of a protein. So the hydrophobic amino acids within the polypeptide tend to fold into the inside, and when they do, that starts to create a 3D shape. And the hydrophilic ones tend to go towards the outside because they're going to interact more with the water that's available. So certain types of proteins, we call them globular proteins, because that's a fancy word and we clearly aren't that creative with names. So the globular proteins are tightly coiled proteins that take on a spherical shape or a glob shape. And they have the hydrophobic groups on the inside and the hydrophilic groups on the outside. An important globular protein within cells are what we call the enzymes. And enzymes are catalysts for reactions. They can also provide support and regulate transport in and out of the cell. But enzymes make sure that chemical reactions happen within a cell that are necessary. So let's just talk about enzymes for a quick second because they are so important within cells. Like I said, they do allow chemical reactions to happen faster in a cell. So any chemical reaction starts with a substrate and then it ends with a product. And most of the time in a cell, those chemical reactions are probably going to happen, but they're going to take a lot of time if they're left on their own. And the reason for that is what we call this energy barrier here. It means that if we, let me give you an example. If we have a solution and we want substrates within that solution to bump into each other, we can add energy to that solution by, for instance, turning up some heat. That makes all those molecules move faster and will bump into each other. So over the course of time, that reaction can happen faster if we add energy to it. What an enzyme does is it decreases the amount of energy needed to complete the reaction. One of the ways that it does that is it has what we call binding sites, and it will bind substrates to itself, and of course that's directly related to the shape of the protein that we talked about. Those active sites are specific to substrates. So if we don't fold the protein correctly, then those substrates can't attach to the enzyme and the reaction can't happen. But if everything is happening properly, then once those substrates attach, then the enzyme helps the reaction to occur, and then a product is released. Now, what can affect this enzyme and how it works? Two really important things that determine how effective our enzyme is at allowing these reactions to happen is the amount of heat and the pH that's involved in the solution. Also the amount of substrate, but we'll get into that later. So if we raise the temperature too high, then the enzyme begins to denature. And what we mean by denature is it starts to break down. So certain bonds within that peptide are going to be broken, and when they do, the shape of the protein falls apart and that active site is no longer the same. Therefore, the substrate doesn't fit in that active site anymore. So it's important that we maintain what we call homeostasis. We've talked about that before, which means keeping the heat and the pH within a certain range, what we call the optimal temperature range and the optimal pH for our particular enzyme. Lastly is the quaternary structure of the protein. So all that means is taking multiple tertiary proteins and binding them together to make the quaternary structure of proteins. Hemoglobin is a great example of that. It has a couple tertiary proteins in it, but they have to stay together, and that helps to transport oxygen throughout our blood. So the folding of proteins in all kinds of shapes is extremely important because that shape determines the function. And the folding of it correctly is so important that the cell also makes what we call chaperone proteins. And chaperone proteins help the proteins to fold correctly. Anywhere from the secondary all the way up to quaternary structure, they assist in folding the protein. And in fact, we've learned somewhat recently that they not only help in folding correctly, but if there are any errors, it helps to undo and redo the folding to make sure that it's correct. So just a quick review. Our levels of protein structure are primary, which just involves that linear group of amino acids all put together. Our secondary structure, which can either be a beta sheet or beta pleated sheet, or an alpha helix. And then we move on to tertiary structure, which means we start to have that 3D effect, and then putting multiple tertiary structures together into a quaternary structure. So those are our 4 levels of protein structure. Okay, so our 4th biomolecule category are nucleic acids. And these are made up of carbon, hydrogen, oxygen, some nitrogen, and also phosphorus this time. And these nucleic acids are composed of monomers called nucleotides. That's similar to our proteins that were made up of amino acids. Our nucleic acids are made up of nucleotide monomers. And their function, particularly within the cell, is to store genetic information and to act as coenzymes and in order to help metabolism proceed as necessary. And then it also acts as an energy storage molecule. And there are 4 types. There's DNA and RNA, and those are going to be your genetic information storers. There are coenzymes, and then ATP, which is the main molecule that stores energy and is used as kind of an energy currency within the cell. Okay, so like we said, our nucleic acids are made up of our nucleotide monomers. And each nucleotide has a pentose sugar, which means a five-ringed sugar, a phosphate group, and a nitrogen-containing base. So let's start looking at our DNAs versus RNA. So our DNA has a pentose sugar. That particular pentose sugar is a deoxyribose. If you need to look at that, you can go back to the slide where we were talking about our carbohydrates and we looked at our monosaccharides, and I showed you the example of deoxyribose. And there's also ribose on that same slide. So RNA is made up of the pentose sugar ribose, whereas DNA, just as the name implies, deoxyribonucleic acid is what that stands for. So deoxy, meaning without the oxygen on the ribose. So our DNA also can contain any of the following bases. They can have a cytosine, a thymine, adenine, or a guanine. Our RNA can have all of those same things except for the thymine. It cannot have a thymine. Instead of that, it replaces it with what we call uracil, and that's the base section. DNA is composed of 2 strands, and they tend to twist into a double helix, whereas RNA is single-stranded. So this is just a closer look at our nucleotide structure up here at the top left. So here is our phosphate group, our pentose sugar, and then our nitrogen-containing base. And for our pentose sugars, here we have ribose for RNA and our deoxyribose, notice that the oxygen is missing here, for our DNA. Now, one thing I do want to point out is that our nitrogen-containing bases are here on the bottom half of the screen, and they fall into 2 categories. There is a, you can either be a pyrimidine or a purine. The pyrimidines only contain a single ring right here, okay? And our purines have the double rings. That's how you tell them apart. And I will say, I am going to have you memorize those 5 nitrogen-containing bases. And my reasoning for that is when we start getting into what the function of the DNAs and RNAs are, I want you to be able to recognize what each of these is, because especially as you progress in the sciences, these become more and more important. So whenever you get a chance, maybe do some flashcards of cytosine, thymine, uracil, adenine, and guanine. So there is complementary pairing between these nitrogen bases, and as they come together, they are held together by hydrogen bonds. So the 2 strands of DNA that come together to make that double helix are held together by the pairing of these bases. And the bases always come together, one purine with one pyrimidine. So not only memorizing what the structures of those 5 bases are, you'll need to know what the complementary base is that goes with it. And that is pretty easy. That'll come naturally after a while. Our guanine always matches with our cytosine, and then our adenine can either go with a thymine or with a uracil. So they come together, like I said, in hydrogen bonding, and that's how they pair. For now, I'm going to skip the coenzymes. We will talk about that later when we get into how enzymes function within the cell. And for now, I'm going to move on to ATP. This is something you have heard a lot. It stands for adenosine triphosphate. And of course, that shows up in Our chemical structure here, we have an adenine, which is a nitrogen base, our ribose sugar to hold them together, and our 3 phosphate groups. These last 2 phosphate bonds here are very unstable and they are high in energy. And the cell utilizes that. So anytime it needs to perform a function that requires energy, which is pretty much anything we do, it breaks apart the ATP bond by hydrolysis. So we go from ATP to what we call ADP, which is adenosine diphosphate. And when we do that, it releases energy that can be captured and used by the cell. So ATP is what we call the currency of the cell. The cell has to pay in ATP to accomplish any function. If you'll remember, when we talked back about carbohydrates, we talked about glucose, which is our monosaccharide. We ingest glucose or sugars, and the carbon-to-carbon bonds that are there are broken down and release energy. That energy is stored in the cell as ATP. And so this ATP is then used as the currency of the cell for ways to pay for accomplishing any function within the cell. So it's extremely important. So now we have gone through all 4 types of biomolecules. I know that was a lot to take in. All very important once we start talking about the functions of the cell to know what those molecules are composed of and what they do. So meet me back here next time and we'll talk again.